The Senate Appropriations Committee is responsible for carrying out the Senate's part in deciding how federal money is spent, as is outlined in Article 1, Section 9 of the United States Constitution. Decisions and recommendations by this committee have helped to lead to incredible scientific achievements, including, but not limited to, landing man on the moon and sequencing the human genome through the Human Genome Project. Heredity has been of interest to man from very early stages of human history. In fact, according to research by Gabriel Rangel from Harvard University, humans have been modifying genomes for nearly 30,000 years. For most of that time, they used breeding programs to increase the prevalence of desired traits in various organisms. Modern science has, however, made it possible for researchers to alter genomes and study heredity in the laboratory environment, to experiment with the genome and introduce any new trait that they wish to observe. The inventions of the polymerase chain reaction to synthesize copies of DNA and the CRISPR-Cas9 system, which allows alteration to the genetic makeup of living cells, have revolutionized the field of biology and allowed new avenues of research. In Editing Life, a conversation with Jennifer Doudna, co-discoverer of CRISPR-Cas9 gene editing technology, an interview by the Lafayette College Policy Studies Department with Jennifer Doudna, a professor and researcher at the University of California, Berkeley, published in May of 2017, Doudna expresses her belief that techniques like the CRISPR-Cas9 system, an invention which she is credited as having been a part of creating, could be used to cure genetic diseases, but that more research is necessary before these cures can be achieved. Concerns do exist about the widespread use of genetic editing. Nonetheless, the Senate Appropriations Committee should expand funding earmarked for research into genetic editing to strengthen American national defense, fight devastating diseases, and bolster the economy of the United States. Doudna, in the 2017 book that she co-authored with Samuel H. Sternberg, Kraken Creation, Gene Editing and the Unthinkable Power to Control Evolution, says, "I could scarcely begin to conceive of all of the ways in which our hard work might be perverted." This quote lays out the core of the controversy surrounding genetic editing. Some of the concerns around the modification of the human genome are laid out by the National Institutes of Health in What Are the Ethical Concerns of Genome Editing? Amongst them are issues of economic differences, limiting whose children could receive life-saving genetic therapies, the potential ability of the wealthy to pay for the creation of their ideal child, the inability of a human fetus to consent to treatment, the moral and religious conflicts around scientific testing on human embryos, and safety concerns, focusing on a phenomenon known as off-target mutations. In 2017, researchers were, for the first time, able to modify the genome of a human embryo, successfully eliminating a harmful genetic sequence. This research is, however, concerning to some because of lab results showing a high rate of unintended mutations in other sections of the genome. These results are concerning because this could lead to the creation of new, dangerous mutations that could be passed on to future generations. Ultimately, the concerns of Jennifer Doudna and the dilemma laid out by the National Institutes of Health highlight real concerns, but could be limited by restrictions laid out as requirements for receiving federal dollars to conduct research. Concerns exist in the alteration in the genetics of crop plants and animals raised for consumption as well. In 2008, Teresa Phillips, who holds a PhD, wrote a paper for the scientific journal Nature, outlining potential concerns surrounding genetically modified products for consumption. One concern she highlights is the risk to the environment where these products are grown or raised, and the potential to destroy the soil. Further, she lays out issues surrounding human health, including introduction to new allergens and the possibility of allowing bacteria, which can take up DNA from their environment, to be exposed to genes that would give them resistance to antibiotics. While these fears should be taken seriously, more research could lead to improved products that address the concerns of Phillips and others who share her views. On February 9, 2016, then Director of National Intelligence James Clapper highlighted another concern around the rise of the use of genetic editing. Clapper, in testimony to the Armed Services Committee, listed 6 of the most legitimate threats concerning the development of weapons of mass destruction. These included North Korea's rapid march towards nuclearization, drastic Chinese military buildup and modernizations, Russian violations of weapons treaties, chemical weapons held by the Syrian and Iraqi regimes, Iranian cooperation with the West to potentially preserve their nuclear capabilities while raising revenue, and finally, genetic editing. Clapper cites the potential of advanced methods of genome modification to be used to create devastating biological and chemical weapons. Increased funding for research into genetic engineering could help combat the potential risk of biological warfare and bioterrorism. The power to introduce new genes into bacterial cells could lead to the production of new toxins and increased resistance to known antibiotics, and could have massively deadly consequences should the public ever be exposed to such a bacterial vector. However, researchers at the University of Buffalo have recently announced their success in programming E. coli bacterial cells to produce antibiotics which the team has used to kill variations of bacteria normally resistant to antibiotics that are currently in use. Developments like this could be used to strengthen the Strategic National Stockpile, a massive collection of medical supplies and treatments funded by the Centers for Disease Control and Prevention, a branch of the United States Department of Health and Human Services. According to the CDC, the Strategic National Stockpile is meant for use in large-scale emergencies to rapidly expand the abilities of the healthcare community. Strengthening the Strategic National Stockpile would help ensure American readiness for disaster, whether it be natural or man-made, and could be possible through increasing the number of grants to university and private labs, as well as increased funding for the Department of Health and Human Services, emphasizing the CDC and the Strategic National Stockpile would be effective countermeasures to the threat of biological agents. Genetic editing and genomic research could not only expand the varieties of antibiotics available for use in the healthcare industry, but could cure many incurable diseases. In October of 2017, the United States Food and Drug Administration, or FDA, voted to allow for further trials of Luxturna, a treatment for a rare form of blindness. In July of 2017, a study funded by Spark Therapeutics, the company behind Luxturna, was published in The Lancet. The document details the use of artificial DNA strands that, when introduced into retinal cells, allowed for the return of patients' vision. In this study, doctors who carried out the initial trials reported patients retaining this improved vision for 3 years at the time of publication, without any major side effects. Gene editing has also been applied to the fight against human immunodeficiency virus, or HIV. A team from Temple University has published their findings detailing their use of genetic engineering, specifically the CRISPR-Cas9 system, to eliminate the DNA of deadly HIV virus from host cells in living animals. This treatment, with further research, could be used to save millions of lives in the United States and across the globe. Writing more money for this type of research into the budget could impact millions of lives. Recent research has led to greater understanding of the potential uses of gene editing technologies in healthcare. Cancer researchers have an intense interest in gene editing. The FDA published in August of 2017 a press release entitled FDA Approval Brings First Gene Therapy to the United States, announcing the unanimous vote to allow the use of genetic editing to treat a specific variety of childhood leukemia, a treatment that pharmaceutical company Novartis is calling Kymriah. The release notes potential dangers of the treatment that genetically alters immune cells of a patient to attack and kill cancerous cells, but suggests that with careful application of the treatment, these dangers could be lessened and the survival rate for the cancer should increase. In October of 2017, the FDA announced its approval of a similar treatment, this one meant for a variety of adult leukemia, produced by the company Gilead, named Yescarta. In Promises and Limitations of Nanoparticles in the Era of Cell Therapy, example with CD19 targeting chimeric antigen receptor, modified T cells, an article published in the International Journal of Pharmaceuticals by Helene Jacobowski, reviews numerous weaknesses of this type of treatment, suggesting that its future as the cure for cancer is not yet solidified. While this may not be the perfect solution, further investment is necessary to improve its life-saving potential. Major changes in healthcare, like the use of treatments involving genetic editing, not only have the potential to drastically change patient care, but also to have massive impacts on the economy. According to data released by the Centers for Medicare and Medicaid Services, the United States spent $3.3 trillion on healthcare in 2016, a figure which, according to their study, accounts for approximately 17.9% of the United States' gross domestic product. The agency reports increases in government spending on healthcare, as well as in out-of-pocket costs to private citizens, and continues on to predict a growth in healthcare spending of approximately 5.6% a year for nearly the next decade. Cancer alone is massively expensive. We're in the middle of a cancer drug innovation gold rush, but it has one major side effect. An article written by Lydia Ramsey and published by Business Insider on June 11, 2016, cites studies by an independent firm that estimates that in 2015, $107 billion were spent on cancer treatments, and details the addition of new expensive drugs and treatments, adding to the cost of cancer, estimating that by the year 2020, the nation will spend roughly $150 billion on cancer treatment. The introduction of new drugs and treatments to the market will create competition and, therefore, lower costs for families and the government alike. This will allow for an increased level of the population to live healthier lives and, in that way, also lower healthcare costs. This will boost the American economy and strengthen the middle class. Increasing government funding for research into new drugs and treatments involving genetic engineering will help to advance research at a faster pace and ultimately lead to decreased healthcare costs. Another economic boom that could come from increased government funding of genetic engineering is in the energy sector with the creation of biofuels. According to the United States Department of Energy, a biofuel is any variety of energy source that comes from cells or a collection of living organisms. The oil company ExxonMobil invested $600 million into a partnership with Synthetic Genomics and recently announced that the team has used genetic engineering to double the amount of biofuel produced by a single algal cell. More discoveries like these could lead to an energy revolution as the world searches for cleaner, renewable energy sources. American biofuels have the potential to interest international investors and to decrease gas prices while increasing American energy independence. American renewable energy is a growing industry, and the potential for widespread use of biofuels is immense. Further research into work like that being done by ExxonMobil and Synthetic Genomics has the potential to bring about an American energy revolution and contribute to growing the United States economy. International trade is a major contributor to the American economy, so increasing trade income would help to expand and strengthen the economy. According to a report from the United States Department of Agriculture's Economic Research Service, released in 2015, profit from the export of agricultural products was $133 billion. The Economist, in its article Milking Taxpayers, which it ran on February 12, 2015, estimated that in that year, the government would spend roughly $20 billion on farming subsidies, which they assert mostly go to large corporate farming operations, to keep steady the profits of American agricultural products amid fluctuating prices in foreign markets. The creation of crops that are more efficient to grow would lead directly to a decrease in production costs and therefore an increase in profits for farmers on large corporate-sponsored farms as well as for family farms. Increasing spending on research for the production of genetically modified crops as well as their impact on human health would expand the American economy by creating greater competition in markets at home and abroad. Despite the existence of controversy, genetic engineering has the potential to strengthen the country. The national security of the United States will be stronger if more research into genetic engineering is conducted, and especially if that research results in the growth of the strategic national stockpile. Modern techniques of genetic engineering are producing amazing new treatments to diseases whose devastation will hopefully soon be left in world history, as the FDA works to continue to get more of these new products on the market. This revision to the healthcare industry can work as a booster to the American economy, lowering costs and saving lives. American energy independence could also, in part, become a legacy of humans harnessing the power of genetic engineering to create new sources of energy and expand efficiency of biofuels. American economic growth from this production of new, cleaner fuel sources by biological means would create jobs and attract foreign investment. The production of genetically modified crops, shown to be safe for human consumption, would increase American competitiveness in markets at home and abroad and will contribute to the growth of the national economy. Innovations in science will continue with or without the United States. However, increased government spending for research on genetic engineering will strengthen the country and benefit the American people.